Hi there, I'm Dr. Ben Britton, and I'll be delivering this lecture on scanning electron microscopy. I'm a reader in microscopy and metallurgy in the Department of Materials at Imperial College London. First off, there's some great resources. This lecture is a very short taster that will give you some of the underlying ideas, principles, and opportunities available with electron microscopy, scanning electron microscopy in particular. I'd strongly suggest that you have a look at Erdmann and Bell, scanning electron and ion microscopy. There's some great online resources to teach you about microscopy using virtual tools, the virtual SEM from the Australian microscopy community. And then there's a great book, Scanning Electron Microscopy and X-ray Microanalysis, that's also fantastic to use. So I like this very short video that sort of gives some flavors of how scanning electron microscopy is useful. So in this video, we can see effectively some three-dimensional images as we scan and rotate our sample underneath the electron microscope. Now, as we do this, we can see fantastic depth of field of features. We can see a range of different length scales, magnifications. It's a pseudo-color image, but it still shows us that we can see complexity that we can imagine with the human eye, but we can see detail that's much, much finer. Some of these features can be seen, and they're down to almost sort of nanometer in length scale. This is well set up to take a series of images and create these videos by changing magnification and object with respect to the microscope. And many of you may do this, for instance, as you look at, say, biological systems, as presented here, but also perhaps as you look at things like fracture surfaces or other larger objects. And some of you may be just looking at fine features, but use this opportunity to pan in view to find and locate the interesting objects that you've got within your electron microscope. Now, in certain cases, we may do sort of in situ measurements. Here we can see actually the electron beam is dehydrating and burning samples, and so those fibrils are actually moving. As we're looking under the electron beam, they're dehydrating and moving with respect to what's going on. These are really fantastic videos to highlight the opportunity afforded by the scanning electron microscope, where we can see a huge range of depth information, and we can start to piece together stories about the objects that we're characterizing. What we do have to remember is that every single pixel within this image is information, and we'll see why that's important as we characterize the information we're collecting associated with the electron-matter interactions themselves. This enables us a whole range of different samples to be inserted, observed, characterized, understood, and to tell stories about the materials that we're interested in. And this enables us to have a huge range of opportunities to look at that point-specific information and gather data. Not just images, but actually data that we can quantify to tell us about, for instance, the size distribution of features, the local chemical signals, and much, much more. So you have a look at the videos associated with nanoflight. It's some fantastic examples to highlight opportunities afforded by electron microscopy. So, why do we use scanning electron microscopy? Scanning electron microscopy is fantastic because we can use both the electron behavior and effectively we can use the electron behavior both in terms of thinking about it related to waves or thinking about it with respect to particles. In the scanning electron microscope, we can look between something in some microscopes of 50 electron volts up to 35 kiloelectron volts. That choice of the incoming wavelength has a huge range of benefits. Not only does it change how surface sensitive the technique is, but it can also change the character of different signals that we see. And we'll see that in a few examples. Specifically, we'll see later in the EDX lecture how that can be used to understand different features at different layers in our microstructures. The wave behavior of the electrons can be useful in the formation of different images and also the generation of characteristic diffraction patterns, and can also be important in certain microanalysis. Considering the particle behavior, that is very useful because we can think about the electrons as effectively sort of physically bombarding into our material, and we can think about what that bombarding does, the scattering, and then the release back of, say, the primary electrons back to give us a backscatter signal. In the scanning electron microscope, we're going to scan the electron beam across our samples. This means we can look at relatively large samples, several millimeters, in some cases even bigger. And we can dial down to very, very small imaged regions, often of the order of, say, hundreds of nanometers in certain cases. There is relatively easy sample preparation. We can do this at near atmospheric pressure in some microscopes, but often we're doing it at a low to medium vacuum system. But we can look at relatively large samples and we can literally stick them on top of our stubs. We don't have to prepare, for instance, very thin and complicated electron transparent samples. We can also, as we scan the electron beam, we basically in an SEM, we synchronize the position of the electron beam with a characteristic signal. And this enables us, for instance, to collect a variety of different signals at each point. And that is the basis of microanalysis. And we'll see that as some examples in this lecture, but we'll see that in much more detail in, for instance, the energy dispersive X-ray spectroscopy lecture. So the basics of the scanning electron microscope is that effectively we scan a probe across the surface, we raster, and at every single point we take a signal, and that signal is used to sequentially build up a picture of our sample. And we just repeat this multiple times to gain the pictures that we've got. Now, what's important is that effectively at each location in our microstructure, we collect a signal, and then it's the separation of those locations that describes the step size of our image. And the magnification effectively is how much do we step within our object to how much do we step within our image. The magnification is subtly different. For instance, you could imagine the magnification, the physical feature size to the observed feature size, to be different depending on whether you're watching this video on a mobile phone or on a full laptop screen. So you've got to be slightly careful about what that magnification number means. What we like to think of more precisely is what is the characteristic resolution of the technique, i.e. how can I tell the difference between a signal at point A to point B? We'll discuss that in more detail shortly. Broadly speaking, this is of the order of 1 to 2 nanometers for most modern electron microscopes. Another very important factor is the idea of spot size. So spot size is basically how representative is the point that you're scanning in the microscope to the area that it represents. And that's a really subtle but important point. So broadly, if the point here, if you've got a material, you've got to consider what is the likelihood of hitting a characteristic average signal that describes the totality between point A and point B, the totality of separation between the stepping of our raster scan. The convenient method to get this right is to try and match the spot size to the information that you're accessing, i.e. match the spot size or the condenser lens settings for the raster that you're going to do. When you want to consider resolution, there are a variety of different methods to do this, but one classical method is to look at effectively a high Z number material on a low Z number system. So this is a gold on carbon, where you have sputtered very small spherical objects of gold that are on a carbon-based film. And the resolution is often how can you distinguish one blob from the next? And if you can do that, that effectively is how you qualify the resolution of the electron microscope. When we scan our electron beam across our sample, we talk about the characteristic signal that we detect. Broadly, we input a focus probe, often a convergent focus probe of electrons, onto our material. That is a high energy probe that is highly penetrating into our bulk sample. The signals that we can characterize, we have effectively a low energy signal associated with the promotion of surface electrons from our subject, and those very low energy promoted electrons can escape and be attracted and collected, and they're called the secondary electrons. And that's the common imaging mode that we'll often use in the microscope. In some microscopes, we'll also get characteristic, we can measure the energy of those electrons, and they can be at slightly higher energies, and we'll see them in a spectrum later. They're associated, they are the Auger electrons in our system. Now, in addition to these, these electrons, these very low energy electrons are typically located very near surface, and broadly speaking, they are from the direct impinging primary electrons. So often they have a very good spatial resolution as determined by how well we can focus the initial probe of our electron beam. Now, the primary electron beam will not just go and create these secondary electrons, it will continue rattling into the system. So it will effectively go into the material and it will scatter as you get primary electron beam matter interactions. Eventually, some of these primary electrons can actually be bounced back out of the sample, and they produce what we call the characteristic backscatter electrons. And these are varying from the spatial source of the initial to tens to hundreds of nanometers away. And so the resolution of backscatter is typically less good than the resolution of the secondary electron image. In addition to the backscatter electrons, i.e. the primary electrons coming back, we can also generate characteristic X-rays. These characteristic X-rays are generated, and they're principally generated by effectively the primary electron beam going into the material. What will happen is it will promote the electrons inside the matter of the material, to promote the electrons from a core state up to a high energy state, and then those high energy electrons will fall back down through a characteristic X-ray transition and as a characteristic energy transition, and in that process they will release an X-ray of a specific energy for that elemental species. In doing so, that effectively reduces the energy of the primary beam by that characteristic promotion of the core electron. And so you effectively, you can only excite the characteristic X-rays depending on the Z number or the energy of the characteristic transitions in there. And the only way, we'll see this in the EDS course, but we'll see that we have to actually capture or grab those characteristic X-rays at the end of the thing. And we'll see why that's important later on. But broadly speaking, effectively, they can come from about 1 to 3 microns, depending on the Z number of our system. And there is significant lateral scattering of our electron beam in our system. And so the resolution for X-ray analysis is significantly larger than the resolution for the secondary electron image. So imagine that we have that focus probe. Well, how do we get to the point that we have the focus probe? And so this is where we require ourselves to have a relatively reasonable ray diagram. It's the SEM was effectively generated off originally a transmission electron microscope, but it has a lot more synchronization and a few more bits and pieces. But very broadly, we start with a characteristic electron source. That can either be a thermionic emitter or something like a field emission gun. That source effectively spreads electrons off the source. We have a condenser lens effectively that will cross over that spreading source into your condenser point. You have an aperture located up here that can effectively reduce some of that spread, and therefore you get your initial source spot size. That can go through a second condenser lens, and then you can have your OL, your objective lens aperture, and that's going to tell you the probe that comes off and down the column. Now, by changing the strength of the condenser lens, we can change effectively the number of electrons that are scattered down into the microscope, as well as the divergence of the source at this point. If this is well aligned, such that we have the optic axis running down the sample, then what we can do is we can apply a voltage to our scan coils, and that enables us to deflect the primary beam across the surface of our sample. We can bring that divergent probe into a convergent probe by using objective lens, and so that gives the final focus position of the electron beam on the sample. By changing the strength of this, we change the location where this probe can focus. And in the SEM, that can vary often between a few millimeters ahead of the pole piece, down to of the order of 40 millimeters. And that is phenomenally useful if we want to scan across very high topographical samples in our microscope. Now we can have, in addition to the focusing of the electron probe, we can now, in our large chamber system, we can have a series of different detectors, such as something like energy dispersive X-ray spectroscopy, cathodoluminescence. We can have a backscatter electron detector, which effectively this is a below the pole piece. And we can have a secondary electron detector. We'll see how that works. So we can hoover up those low energy secondaries. And we have our relatively large sample or a series of different geometries and other bits and pieces where we put the sample and the detectors inside the chamber of our system. What's important is that you have a rough idea about how this column is set up and how that matches the physical column of our microscope. So just to remind ourselves, we have a few different sources that we can use to produce our primary electron beam, and that will change the character of the energy characteristics of the probe. So the cheapest form of source is a tungsten hairpin, very similar to an old-fashioned light bulb. We effectively have a low work function material that is heated to relatively high temperature. effectively by applying a current through it. And then we can rip off the surface electrons by effectively having an anode just below this. We can have the similar setup, and now we have a lanthanum hexaboride, an LaB6 crystal, that effectively has some slight benefits with regards to the size of the initial source and also the probe current. And many of our microscopes in the Department of Materials here at Imperial, we have what we call field emitter sources, where effectively we have very, very sharp sources that have high brilliance. So that's a high number of electrons per unit area, and that's very useful in order for us to capture our signals relatively quickly, and also have a high spatial resolution because the initial probe size is relatively small in our microscope. Now, there are a range of different choices of these materials. We have the thermionic tungsten, the thermionic lab6. We have two different FEGs, the Schottky FEG and the cold FEG, and there are a series of different properties of these different parts. So specific things that may be interesting effectively is the energy spread of the electrons, because that can of course influence things like the chromatic, i.e. the different energy chromatic aberrations associated with the different energy of the probe. It can also vary the total probe current, which can be important for microanalysis, but it can change. We've also got to be careful that effectively this probe, although it has a lower maximum probe current, although these numbers have gone up these days in modern instruments, the brightness, how that can be focused into a single spot, can actually be very, very high from a Schottky field emitter, whereas it's much more difficult to get that from, say, a tungsten field emitter. Other things are effectively how stable is the source over time. So the Schottky versus cold, most of our instruments are often Schottky instruments because they do not drift very much over time. The issue is that with a Schottky instrument, you often require a relatively high vacuum system. There is a relatively good life, which is good for the system, but the one item that's not included on this list is that a Schottky source is around something like £30,000 to change, whereas the thermionic emitter is about £8. So this you've got to look after better and avoid causing damage to it, and so that's why you often require more training, and you have to be more careful with your samples if you're using a Schottky field emission microscope. So we've now got our electron source, we chose the appropriate source, we then can consider what the condenser lens and aperture is doing, but effectively in this case we're going to choose the effective probe current. So imagine that we have a fixed current off our source that's being pulled by our anode, pulling it down into the microscope. If we change the convergence of the condenser lens, that effectively changes the position of this crossover and changes effectively the number of electrons that are going through our aperture. This changes effectively how parallel the beam is coming through. So we can see this is a divergent source where it has a low convergence. This effectively is a convergent source with no crossover in our system for this example. And we have our aperture strip, which effectively is then selecting out the total area of that primary beam that we're using. So this is why the aperture can change the total current and the total spot size. And in some microscopes, you can also change the deflection associated with the focal point on the condenser lens system. So this effectively, the OL aperture and the CL setting will change the spot size and probe current. And depending on the microscope manufacturer, they will name this two different things in the system. And some microscopes, Zeiss instruments, for instance, will just have a setting that will deflect the beam through the different apertures. Joule instruments, for instance, or FEI instruments, they'll actually physically change the aperture by moving it through the system in these cases. But fundamentally, this broadly, it changes the spot size and it changes the probe current of our system. Now the objective lens is now going to change effectively this sits below the aperture strip, and this changes the focal position of this beam that comes out of the aperture. So we can now choose, does it focus high or does it focus low? So here we've got at focus where the focal point is directed on the sample. We can have over focus where the focal point is above the sample, and we can have under focus where the focus is below the sample. Now, what's important is you want to choose the focal point to maximize the information that you want to look at, and often you'll want to direct this to have the focal point located on the sample. Now, we can have some choices about, do we move the sample upwards and focus at a low, or do we move the sample down and focus at a lower point? And there are some specific benefits. So often, provided you do not hit the pole piece, which is an expensive thing to do, but if you do not hit the pole piece, often going close to the pole piece gives you a high magnification. In contrast, Going further away from the pole piece reduces the magnification, but often it improves the depth of field. Why does this matter? Well, you effectively have what we call a region, which is the zone of least confusion. So this is a region where the spot size matches the step size in your grid. So if you've got this well set up, you've got the point of crossover, and you now have that you've matched the step size here and the step size here. And so anything between these 2 points effectively is in focus, so it's as sharp as it can be. If your feature and your step size was wrong, then you can imagine that your feature is located smaller than the size of your pixels, but the neighboring points effectively are sampling the feature twice. And that's effectively how you get blurring, so your resolution is limited because you've chosen the wrong focal length and wrong pixel size for your problem. So you have this region of the zone of least confusion where your features are equivalent to your step size in your system. Now, hopefully, you can see this is a relatively simple piece of geometry where you have the opening aperture size that effectively determines, together with the point where crossover happens. You can see there are a pair of similar triangles with the opening angle alpha that will control effectively what the effective working distance is for an appropriate size of feature over here. Now, what that means in practice is that effectively a long working distance gives you access, because effectively you have, in the long working distance, this angle is smaller. This means effectively the zone of least confusion is longer, so the depth of field is better. For a short working distance, you can see effectively this means that the angle is wider, the convergence angle is drawing it in much wider, and therefore for this particular example, the zone of least confusion is narrower, so the depth of field is narrower in our system. Now, we note that electron optics are not perfect, and broadly the typical analogy that we give is that if we were to compare a magnifying glass to the electron optics we have in our microscopes, it would be equivalent to looking down a beer bottle. Now, the ultimate resolution, how much we can distinguish 2 points, depends effectively how well we can focus that probe into a sharp point. And there are several aberrations that can be present. We have the first order chromatic aberration, which is dependent on the energy spread of the beam. We have the third order spherical aberration, which is related to what's called the disk of least confusion. And we can also have, and you'll see what this means in a minute, but we can have an astigmatic probe, where effectively, although we would like to have a nice needle point pinprick, we will have something that instead of having a spherical cross-section, circular cross-section, will effectively have an ellipse. And so that's astigmatism in our image. There are some relatively simple equations that describe the limiting points of the resolution, and they are additive, that the effective spot size is a sum of effectively the diffraction-based error, the spherical aberration, the chromatic aberration, and the ultimate Gaussian spot size. The key take-home from this is that there is a strong dependence on the convergence angle alpha, as well as how good your microscope is, things like your aberration coefficients, and how good your probe is, your energy spread in this case, and the wavelength of your electrons as well. What this means effectively is there is a specific sweet spot where you can have an optimum convergence angle to get your best step size. And you can see effectively how that's the summation of the diffraction error, the spherical aberration, and the chromatic aberration. And we can see that this is broadly, each of those terms depend on the convergence angle themselves. For more information, you can see chapter 7 of Erdmann and Bell for this. So now we've got our very sharply focused probe of electrons that's going into our system, and we can now effectively consider the signals that are generated and what the images will look like through the consideration of those electron-matter interactions. So we have to think about what is happening in our electron-matter interactions. So first off, we've got the generation of those secondary electrons, and typically that is effectively very similar to the focal beam width, or how spread that beam is as it comes into the sample. So that's all about your focus, and it's coming from the sort of top few nanometers, and the spatial resolution is somewhere 10 nanometers or less. And of course, it depends on the emitter type. So a tungsten lab 6 instrument is of the order of 10 to 100 nanometer spatial resolution. A FEG instrument can get better up to about 1.2, and some can be slightly better with some tricks. Because the primary electron beam will scatter before it generates backscatter electrons, effectively the backscatter signal is the convolution of all those scattering events that gives you a spatial resolution, depending on how you set it up, of somewhere like a micrometer or 1 to 2 micrometers for your backscatter signal. There are tricks to get that better, but you have to be very careful about this. Depending on your material, then the X-ray and CL generation will happen in the larger interaction volume, and that will often be something like 2 to 5 micrometers for where the total signal is generated. Now the probe size, that's, as I briefly mentioned, is dependent on the emitter. So a tungsten filament has the largest probe size, a Schottky field emitter has one of the smallest, and a cold FEG is slightly smaller. Broadly, it's also voltage dependent, so a higher KV will often have a larger probe size, and that's often why some people, for very specific high spatial resolution work, may want to use a lower KV for imaging. And actually, for the majority of our FEG instruments, for imaging, you'll often run them at 5 keV. They're optimized for those setups. Now, of course, the spot size, that's effectively related to the aperture and condenser lens, how you've converged the beam. That will also change the probe size. And of course, all the aberrations in the microscope, as we've seen, will impact those. And generally speaking, if you spend more money on your microscope, you get fewer aberrations because they have made it better with some of the technologies that are involved in forming the probe in those systems. Now, if your primary electron beam is going into your sample, you can look at the characteristic generation of X-rays off the surface of your sample. And so you have a very strong, so the number of electrons as your y-axis, the electron energy as your x-axis. You have a significant generation of those secondary electrons. Your backscatter electrons are broadly those electrons which are greater than 50 electron volts. And we have characteristic Auger with specific energy transitions associated with the characteristic energies of our system. And so that's why we can do chemical analysis based upon Auger electron spectroscopy. It's very good for surface-based analysis. The backscatter electrons, effectively, we can have directly elastically reflected electrons, i.e. they are the same energy as they come in, or we can have those associated with plasmon losses, so we have literally rattled the lattice very slightly, and we kick back the primary electrons, having lost transitions associated with those primary electrons. And that's up here. But the backscatter signal, broadly in most backscatter signals, effectively is counting all of the electrons from about 50 electron volts up to the primary beam energy in our system. And this is described in a lot of detail in a fantastic book by Riemer. So the common imaging modes, we've seen this before, that the secondary electron mode is fantastic to image the surface of features and gives you very high contrast associated with topography in our system. As I've said, this signal is generated often from the electrons 1 to 50 electron volts, and we'll see how the technology is used, the Evenly-Thornhill detector, to grab those signals. Backscatter electrons, often they are the higher energy. They are effectively the primary electron beam that is reflected back to the sample. And of course, the ability to reflect those signals is related to the topography. You can imagine this like smashing an electron into it's considered like a game of billiards. If you have a flat surface, the billiard will be reflected back into your face. If you have an inclined surface, then the billiard will be reflected off at a different angle. So if you set up your detectors well, you can choose and you can get something called the topo image by looking at the different trajectories of the backscatter signals. Similarly, the coefficient of sort of the reflection, i.e. the backscatter coefficient of your system, can give you access to chemistry. And due to some subtle effects, because we're often imaging something like a crystalline sample, actually you can also get electron channeling, and we'll see what that is in a bit. This is related to electron diffraction, but very briefly, that can give you access to information inside our materials, and that can be very useful, and it's important to consider in a variety of parts, a variety of challenges for materials characterization. So the secondary electrons, as I mentioned, the primary beam goes into a sample, and effectively it promotes core electrons. They escape, and they produce a signal. They are primary electrons that are primary electrons come into the sample. Secondaries are generated effectively as electrons are liberated off your material, and they have to be near surface because these secondary electrons would otherwise be absorbed by the material itself. This is an inelastic scattering process in how this works, and they leave the specimen surface often in any direction. So we have to apply a very slight positive bias to hoover up those secondary electrons into our detector. Now, there are several types of secondary electrons. There's the SE1 electrons. These are generated from your actual sample, and they're generated from the primary interactions of your primary beam, promoting electrons from your surface. The SE2 electrons effectively are that your primary beam has done something in your sample, it's scattered lots, it will then produce backscatter electrons. Those backscattered electrons will then generate the secondary. They are not located at the same position of where the primary beam has promoted for the SE1 electrons, and so they are effectively, they worsen the spatial resolution of your system. The SE3 electrons are those that are generated interacting within your microscope, and so those are effectively any electrons that then are liberated from other parts of your microscope itself. The efficiency of generation of your secondary electrons is proportional to your sample tilt, and as you point it towards your Eberhard Thornley detector, as you point it towards that, you can improve the SE detection efficiency. The ETD itself, this Eberhard Thornley detector, this effectively it's a very clever device where you have a Faraday cage that's effectively have a positive bias that can therefore attract your negative electrons into your surface, into your material. You have a scintillator, so you're converting the electrons you're scintillating to produce light. This enables you to then put the light through a photocathode. That photocathode can count the number of photons, and if you have a photomultiplier tube, you can increase the signal to noise for that system, which means you can generate a lot of signal for effectively a few incoming electrons, which means you can count for a very small period of time. If you read the signal and synchronize that with the primary beam, you can therefore generate your secondary electron detector signal. Now, the creation of secondary electrons is also, well, of course, it's related to basically how well can those secondary electrons escape the surface, and now you can see why it's strongly topographically dependent. If your ETD is located up here, for instance, you will get a significant bright emission of electrons from this location, because there are lots of electrons that can scatter and easily get towards your target detector. In this example, the surface area where the electrons can escape, actually there are fewer that are escaping and hitting your detector up here. So this will be less bright. And if you were looking at this point and you had the detector over in the top right, of course this would look darker. So you effectively get that sort of pseudo 3D shadowing associated by the position of the ETD with regards to the surface topography that's present in this sample. This enables you to see the surface topography, and so this is an image that I captured from a superalloy-based micropillar that I've deformed. And this is really nice because we can see surface roughness associated with plastic slip, and we can also see etching morphology associated with the gamma-gamma prime structure in this nickel-3 superalloy. The backscatter electrons, broadly speaking, you have the primary beam. If it hits the primary nucleus and reflects, there's effectively some small plasmon losses. That's your primary backscatter that has those near elastic scattering events. But you can also get that backscattering due to the interaction with the nucleon cloud, and so it will zoom back, but it's the primary electron that you're signaling. So this effectively will only lose electron energy from the primary beam due to some small scattering as it changes course and comes back. This is very useful because we can see, if we imagine that to our billiard ball type experiments, if the sample is flat, we'll get a high efficiency back to our detector. If we're going to an inclined, it will bounce back at an angle. And if you therefore have effectively a segmented detector, you can look at the differences between the left and right quadrants, and that can give you the idea, is it pointing this way or is it pointing this way? And so that enables you to get the topography information of your surface. If your material is relatively flat, the efficiency variation of your backscatter signal is broadly, the first order efficiency is related to the characteristic Z number. So how efficient is the material at backscattering the electrons back to your detector? So that's the topo variations. This will have a higher signal if it's flat compared to the second case, because you have off this tilted sample, the electrons are being bounced in a different trajectory distance. Now, the efficiency of generation of backscatter is both proportional to the primary energy of the beam and the elemental composition. And so, for instance, if you want high contrast of backscatter signal to show between these different species, often you'll run the system at high voltage. so you can get a strong variation that is broadly proportional to the Z number of your system. It is also a variation, so Z number you can see here in this system, you can see it thrown the other way, but if you look effectively as you change your voltage, you can change the ratio. So depending on which species you want to look at, you can select how to optimize the contrast in backscatter by thinking about it a little bit. And there's a great description of this in Rima's work, in this fantastic series of work, and Rima and Tolkamp as well for this. Overall, you can look at, of course, these different signals as they're just different detectors. And modern instruments, you can have a frame grabber that grabs all the signals of different detectors at once. So this is showing the backscatter electrons. This is showing the secondary electrons. This is showing the topographical information from the backscatter. So that's showing, say, the difference between the left and right quadrants. This is the compositional variation. Now, the question you should have a brief think about is, well, if you've got a material, what would be brighter or darker in composition? What's going to control that brightness or darkness? Have a little bit of a think. Well, a brighter area, that means to have a greater efficiency of kicking the electrons back. So that means it has to have a higher Z number. So the lighter materials will look darker in a compo map. The higher Z number materials will look brighter in a compo map. And if we look at aluminium versus silicon, and I have to peek at my periodic table for this, they are very close to each other, but broadly, effectively, silicon is heavier than aluminium. And so hopefully, if I've got this vaguely right, we can see that regions that are silicon rich compared to aluminium rich will look slightly brighter, i.e. this region, than the aluminium-rich regions. So silicon-rich regions will look slightly brighter than the aluminium-rich regions. And this is a precipitate with slightly different chemical composition. The next thing that's just to bear in mind is that all electrons in the electron microscope will channel. And so this is if you have a crystalline material, they will channel. I like to think of this analogy, if you're driving along and you happen to drive past a vineyard, you will imagine that as you look on axis for the vineyard, in certain scenarios, those electrons or your view will go a long way. And so that means you reduce the signal that gets back out and you probe much deeper into the sample. What's really nice about this for a backscatter channeling image is that effectively as you rock, i.e. you change the angle of the incoming beam as you deflect it across the sample, you can introduce the beam at different angles into your sample, and this creates contrast. The other way that you can rock the electrons with regards to the sample is to rotate the crystal. So if you have a polycrystal sample, different grains have different crystallographic orientations. They present themselves, they can give you contrast associated with channeling. So effectively, if you have the opportunity to rock your beam on, say, a single crystal at low magnification, you will generate an electron channeling pattern. If you can focus the probe and rock about a single point, you can get effectively the selected area channeling pattern. And if you have a polycrystal, for instance, with different grains of different orientations, then you'll have effectively an electron channeling contrast image because you'll have an association of the presentation of the crystals with respect to the primary beam. So the first example I show you is a polycrystalline nickel sample, which is very difficult to etch. So it's quite difficult to see grain structure. And Natalia, who is a master's student, actually got this on a secondary electron image that we get channeling contrast revealing different grain orientations. And you can see these are different, what we call annealing twins in our system that show common colors because they were common orientations and the parent grain over here. And also it shows defects such as scratches in this image. If you're looking at a single crystal, and so this is an example that I grabbed off a large single crystal silicon wafer sample, because I'm looking across multiple millimeters, that changes effectively the incoming angle quite significantly. In fact, I'm looking at a wafer slice that has an 001 axis pointing out of the page. That is the center of the 001 axis. And these are effectively looking at different crystal planes. This is a 110 type plane, and this is a 110 type plane, and then associated other planes, etc., that go through those zone axes in here. These are specks of dust, fiducial markers, as we like to call them in our systems. The benefit of the electron channeling patterns is that they, similar to the TEM, you can effectively choose to image based upon those specific diffraction conditions. If you point the primary beam down that, and the crystal is bending around that, then the contrast will vary according to this specific beam condition. So you can alter the G vector, And that is very useful because you can reveal things like dislocations and twins in microstructures, and this is shown from Marty Crimp's work. This is growing in popularity. It's not impossible to do on a microscope. So the research group I'm leading, we're trying to do some of this in anger, but often you'll get this by accident. If you'll just look at a sample carefully on a single crystal, you can reveal things like dislocations without too much effort in the SEM. This is a really nice example to show where this works in more anger. So if you have a backscatter detector, and this is or forward scatter detector, so the electrons have moved forwards in this case, but they're scattering backwards, scattering forwards in this case. We have 3 detectors RGB. We can get effectively very beautiful images. So actually, this is now published. Space Rocks is the paper name by Vivian Tong, Daniel Gorin, and myself. In this particular example, effectively, we're rotating the sample with respect to the primary beam. We're imaging back to the same position, and we reveal effectively the substructure in this meteorite sample. And we can see effectively lots of small angle grain boundaries, as well as a series of different variants in this FCC BCC crystal structure. In the SEM, we can vary the voltage. So high voltage, the electrons can probe further. Low voltage, we go more surface sensitive, and that can be very useful. There are specific considerations that often if you, for instance, have a charged sample or you have a sample that will beam damage, often you may want to use low voltage so you can reduce those effects. Now, what's really important, if you have a sample that is charging, so it's not pulling the electrons away, then what will happen is you will actually build up a localized regions of charge. So you get a load of negative electrons at the surface. That will deflect your primary beam and can cause some artifacts. And it may also act as a surface lens. So you would be slightly careful if your sample is charging. So charging is a bad thing. And so you'll see as a bit later, we tend to coat samples. That coating of samples enables us to access higher voltage. But we also might need to use the high voltage to actually get through the surface coating. There are a variety of coating systems that we may want to use for lots of different reasons. Very briefly, something like chromium, platinum, or gold are very good because they surface cover very nicely, but they're terrible for microanalysis. So carbon, for instance, is quite good if you want to do microanalysis, because often you'll just see the characteristic transitions from these transition metal elements. Now, the voltage accessible depends, of course, on the emitter type and how the microscope is set up. So imaging, for instance, often on a field emission gun, it will be optimized to around 5 kiloelectron volts. If you want to do, it can go higher, of course. You may go lower depending on the setup. But for a thermionic emitter, often you're limited because you need to pull the electrons off the emitter, and so the emission efficiency is dependent on the voltage. But one of the things I would suggest is that using an electron microscope is actually an opportunity to do direct experiments. And one of the experiments is characterizing what is the best imaging conditions for your particular problem. So you should try imaging the same region at different voltages and see what images you get and see what you like and what information can be revealed. To operate your electron microscope, and this will be it will require a bit of practice and some training, but hopefully you can see this ray diagram. It matches to what the electron microscope looks like. And so different microscopes will look subtly different, but they have the same problems. You have the electron gun at the top. You have effectively gun alignment to optimize that. You have effectively maybe an airlock or something to protect the primary gun. So if you crash the vacuum or ingress air into the system, it can lock very quickly and restrict damage to the emitter. You then have your condenser lens arrangements. You have your objective aperture that you may physically be able to manipulate depending on the microscope. You have your scanning coils that control the deflection. You have the focus of that, the objective lens setup. You have the pole piece. That's physically what you see if you look into the chamber. You have your stage that could be motorized to move your sample around. And you have your sample chamber with perhaps a series of different detectors that are located on the microscope. Now, to do the alignment of your microscope, often I would suggest that you work your way down the column. So you start at the top and you align the gun best as possible, or the manufacturer does that for you. You then align the condenser lens. You then align the objective aperture. You then align the objective lens down here. And you can do this iteratively. So once you've lined it up once for your particular voltage, you can go back to the top and you can nudge it to get it just slightly better. And the whole purpose of this is to draw the primary beam down the optical axis of the system. So you're hitting the sweet spots of each of the alignment features that you want to use later on. And so you'll reduce effectively distortion, astigmatism, and a whole range of other bits and pieces. So the alignment process is that often you'll insert the sample, you'll evacuate, you'll turn on or up the high tension, so you'll turn on the voltage effectively your system. You can physically bring the sample towards your working distance, being very careful not to smash into anything. So you might want to, if you're moving significant distances, you may want to effectively look on an infrared camera, for instance, a chamber scope in the system. You want to find a region on your sample near to what you want to look at eventually, and you want to rough focus. So you want to roughly get it so you can just about see what's going on. And then you want to find something like a circular speck of dust or a high contrast feature. Important that it's not square or rectangular, it's something circular or rough in that feature. You can then do a coarse or fine focus to try coarse and then fine focus to get that best in alignment. And then what you're going to do is you're going to wobble, so you're going to effectively rock the focus back and forth. And specifically what you're hoping to do is to make sure that as you rock it back and forth, I'm just going to show my camera, as you rock the beam back and forth, effectively you're going to do this for the rocking back and forth, apologies. So that's what's going on in the beam, and you want that to go straight down the center. If it's offset, then that rocking will create that this beam will rock about a different pivot point. And so the sample will move lots around that pivot point. And so that's effectively all about this alignment of aperture, is to reduce the rocking about the pivot point. Once you've got that alignment for the gun is going down the column, you then correct the focus, and now you're going to do the stigmatism. Now, a specific point when you're doing the stigmatism, if I'm ever watching you use a microscope, what I really want to see is that you're at arm's length. So your arms are stretched as far as possible, and you're looking as far away from the screen as possible, because it's much easier optically to see what happens. New users, what they'll do is they'll move really close to the screen, and they'll look and they'll try and see this sort of thing, and you'll miss the big picture. So you often want to step a bit further back. And so often as a supervisor, it's very easy to see this because I'm often standing behind and I can see exactly what's going on and I have an experienced eye. But use arm's length microscopy to start off with for these problems. So the alignment, so I've imaged effectively a circular pillar in this example. So if it's well aligned, as you rock and wobble through the focus, effectively it just goes in and out of focus down this bottom right image. If the X aperture is misaligned, then effectively it will rock left to right. So you can see that moving left to right. If you've got X stigmatism, you can see that effectively your image, it's blurring diagonally. So you can see it's going in and out of focus, and that's effectively where the, instead of having a circular probe, you now have an ellipsoid probe, and it's ellipse in the X. So as you rock and you bring the beam in and out of focus, that stigmatism gets convolved on the features that you've got. And the total misalignment will have this mixture of those different alignment steps. Now, those little images look kind of cool, but they're really important in terms of quantitative assessment of features. So if the system is well aligned, you can see these higher order structures at higher magnification on the surface of this silicon pillar example. Even with the X aperture, you can start to see that you don't get these, the sharpness is not presentable at these corners. And if it's horrifically misaligned, you can see you've got blurring because you've got the stigmatism convolving with the features that are present on the surface of your sample. So it is well worth spending time aligning. And specifically, you want to align at a slightly higher magnification in your alternate image, and then you can back off the magnification very slightly. Now, please do note that once you've got the system aligned, you've then got to remember that your images are data. So this is effectively that micrograph of that iron nickel meteorite. It's a really cool image with a huge amount of data that's present. We can see that effectively at each XY position, we have the signal, the backscatter signal of this sample. That's telling us about the number of electrons that are hitting our detector, and it's encoding the first order signal that we've got effectively as we have bright features, and they are I think they're nickel-rich regions. Yes, they're nickel-rich regions. So they are the FCC crystal phase in this example. And they are typically located on the boundaries of the BCC iron-rich phase. The next thing is you can see there is a grayscale variation of the different grains within the BCC. And that's due to the different crystallographic orientations. Now, importantly, this image is grabbed on a detector, which is simply counting the number of electrons that are hitting it. And then we are representing it on our sheet of paper, or specifically our screen. So our screen is effectively a color screen, is what we call a 24-bit image. It effectively has 3 channels, a red, green, and blue channel, each of which have 8 bits. So they can count effectively 1 or 0 to 2 to the 8 minus 1 in red in each of those colors. For a grayscale image, effectively, therefore, we're counting a whole range of gray levels 0 to 255. So from a representation, that's useful to know. But if we want to do some image maths, so we want to effectively classify different regions, throw this effectively to try and do segmentation, we want to optimize to have the best contrast to show those different features. So you can collect images effectively by changing physically on the microscope the contrast and brightness, and you can look and see what features you do and do not see in those things. Specifically, I can show a histogram, and I can do this live on many of the microscopes, but if I grab this well, and I've grabbed this actually in a 16-bit grayscale image for this particular example, and in this capture data of 16 bits, I've got the grayscale intensity, 0 to effectively 2 to the 16 minus 1, over here. I've optimized to have the majority of the data that is stored towards the center, and often you'll want it effectively, the Gaussian peak will look something like this, or maybe just slightly better would be something like this. So the tails of the distribution, the very white regions or the very black regions, are few and far between. If I do this well, it means I can effectively play with the contrast digitally. And so that effectively means I can do stuff like I can augment the contrast and say, actually, I don't care in this particular segmentation here. I want to look at the grain-to-grain variation within the BCC phase. So I can stretch out that central peak to look at that. Or I can segment it to suppress the grayscale variations in this and just look at the locations to binarize for a particle-based analysis. Or I can histogram equalize to give you a good representation with regards to this video. But I can do that in a post-process because I've got good quality data with a good range of information to start off with. I've spent the time on the microscope to optimize the grayscale before I've saved the data and walked away. So have a little bit of a think. You can basically load the data into ImageJ relatively quickly, or on many microscopes, as I say, you can grab the data and store it and grab histograms as you go, and do have a look at the file formats that you're using, and preferably use a lossless, so something like a TIFF-based lossless image, and ideally something like a 16-bit TIFF if you want to grab the full data in this. So capabilities are effectively you can look at a variety of different samples. So this is one example where we're doing some in-situ methods. So this is between a range of us. And I was compressing a series of pillars that I'd focused on being machined. I was compressing the surface. This is a diamond tip. I've got huge depth of field. It gives me a very visual assessment of what's going on. And then if I do stuff particularly well and I can do things well, as I'm actually doing the deformation, I can do in-situ deformation, watch it slow scanning. I can take the sample out in an alternative view. I can look at the features that are present, and I can zoom in and reveal stuff like the slip steps and the interaction of deformation with particular features, such as the gamma-gamma prime structure in this material. If I do this really well, I can also do an in-situ video, and I can actually synchronize this with the external actuation, i.e. the movement of the flat punch onto the surface of my pillar. And so I can see here I've got effectively the load versus displacement, and the material starts to yield. One slip system will activate. It will activate on another slip band. Another slip system will activate here, and there's a third that happens around the back. And I can see and correspond the actuation that I've got with specific load drops, etc., as I progressively deform this object. And this enables me to tell stories, but the specific region here that's really clever is that I managed to optimize the contrast of my scanning electron micrograph, that this is running at a relatively fast frame rate, so I can characterize what's happening against my actuation. Even if you're not running in situ, this means I can grab the data as quickly as possible, which means I can either shorten the amount of time I'm spending on the microscope or look at more samples, both of which are valuable in these problems. If you're based at Imperial College, we've got a whole range of microscopes. Actually, there's more than this list now, but we effectively have a series of tungsten microscopes. There's a couple of joules, 3 joule instruments that are tungsten, and they've got a series of chemical detectors and microanalysis. We have a series of field emission gun instruments. So we've got 3 Zeiss instruments. A new one is about to... sorry, maybe not. 3 Zeiss instruments are currently with us. We have an FEI Helios dual beam instrument that has an SEM. There's a tabletop, and there's another instrument as well called the FEI Quantum instrument. So there's a series of different ones with a variety of different extra things that have different imaging and other bits and pieces and access and things like that. A major aspect of scanning electron microscopy is actually about sample preparation, because if you spend time preparing good samples, you'll get more data. And the first question you should ask yourself is, what is the aim of the experiment? What do I hope to learn? For an SEM, the sample needs to be vacuum compatible and fixed. You do not want to degas the sample. That means effectively release hydrocarbons or gases into the chamber. Your samples can't be wet. They're very expensive vacuum chambers. Each, like a microscope, is 100 to 500,000 pounds instrument, so please don't cover them in crud. Your sample has to be suitably conductive to reduce charging in your system. You want to be free from preparation artifacts. So, for instance, if you're a metallographic person, you want to polish it to remove sample scratches, etc. And it should be suitable for analysis. And you need to think about what method you'd like to do analysis for your sample prep route. So for vacuum compatible, it needs to be typically a dry sample to avoid degassing in your system. So it should at least be sat out for probably a day after you've done the initial prep for most systems. Do talk to group mates or others, or if you need to, talk to the facility managers to ask how they'd recommend sample prep for your particular challenge. You want to reduce contamination, and a major point of contamination is actually your fingers. Your fingers are great in terms of manipulating samples, but the oils will effectively contaminate the surface and contaminate the microscope. And specifically, you'll see this in practice as you scan across a contaminated surface. What will happen is the electron beam will crack the hydrocarbon and you'll build up a black box. It will darken as you effectively do electron beam lithography. You can also get contamination from wet samples, as effectively they degas in the vacuum chamber and coat all the expensive microscope with the crud that was on your sample and mess up microscope sessions for other users. So this is the SEM is a very expensive desiccator. Use something else. For your sample preparation, you need contrast, and specifically, you're either going to look at something like topography, you're going to look at channeling contrast, or you might look at chemical contrast in your system, depending on the method. The electron beam should move across your sample nicely and not produce charging. And a classical charging artifact is that you're scanning across the sample. What will happen is the beam will charge up. It gets to a certain point. So as it charges up, the emission gets brighter and brighter and brighter. What will happen is it will suddenly discharge. The whole sample will shift slightly, and you'll see these line artifacts appearing. So this is a charging eye sample. If it's not charging nicely, you get these beautiful pictures of eyes with no charging artifacts, and you see a lot of detail in your point. So microanalysis charging is really important because you're not getting microanalysis from the specific location you care about. You also get beam deflection, and you get poor images and poor microanalysis thereafter. You can coat your sample to reduce charging. You can use gold, platinum, chromium, or carbon. It depends. Again, talk to your group mates, have a look in the literature, or talk to the facility managers for advice. You want to use a silver paint to ground the sample to effectively enable you to connect your sample to the microscope, and that gives you a good conductive path to ground. And in some microscopes, you can use a charge compensation technique. You can use a flood gun or environmental SEM if you have samples that are charging that you can't use these other methods with. So to summarize, in an SEM, you're raster scanning the electron beam across your sample. You collect a signal from the electron-matter interactions, and you synchronize the detector and beam position to give you information. You have to be careful about consideration of the resolution, of where the signal is generated, how you've scanned and formed the beam. You have to be careful about what you consider with magnification, and this is important to consider the spot size, probe volume, and scan step size. Sample preparation is ultimately very important because the quality of data depends on the quality of sampling that you do. With that, I very much hope you've enjoyed this video, and I thank you for your attention. If you would like to, you can drop me an email, b.britton@imperial.ac.uk if you have any questions about this, and we'll have a Q&A session where I'll answer questions across the 3 topics later in next week. Thank you very much for your attention. I do suggest you have a short break. Enjoy the next video when you have a moment.